Good morning everybody, my name is Rein Brys. I'm working at the Research Institute for Nature and Forest, and the title of my talk is Improvement of eDNA Detection and Quantification via the use of internal positive controls and multiple markers. Although we currently know that the sensitivity of most eDNA approaches is exceeding that of traditional or conventional monitoring techniques, there are still improvements to be made in terms of increasing detection resolution, increase also the precision of our measurements, avoidance of false positive and false negative detections, and finally increase the capacity for absolute quantification, which is needed to predict population densities in the landscape more appropriately. Given the limited dispersal capacity of eDNA molecules in the water column, especially in lentic systems, it is important to take subsamples in an integrated way to increase the detection probability of rare or locally occurring species, which we demonstrated in an experimental setup of caged amphibian and fish species in a pond. And based on these findings, we showed that the eDNA cloud in the water column is only detectable at a few meters from the source, which is affected by the number of individuals in that cage. But on the other hand, we also found that sampling in an integrated way allowed to detect even one or a few individuals at a fixed point in the pond. If we are searching for a rare, protected or invasive species in a certain area, it is important to have knowledge on the temporal eDNA patterns of your study species. For instance, we recently finished a temporal 2-year study in a few ponds that are infected by American bullfrog in Belgium, to obtain insights about the best period in the year to detect this species. In our case, this appeared to be around August, when larvae and juveniles are growing up. Another aspect in which large differences can appear in terms of eDNA recovery is how we preserve and storage our samples. To test this, we created a mesocosm experiment in which we included 3 fish species at variable densities. And after 1 week, we took similar-sized water samples and exposed these to 5 different preservation treatments. The first one was extraction immediately after filtering. The second one was freezing the filters for 8 months. The third and the fourth one was including a conservation buffer, Longmire and Sarkozyl respectively. And finally, we also stored water in the freezer for 8 months, after which it was filtered and extracted. For each of these fish species, we then developed species-specific primer processes to quantify the number of eDNA copies following these different preservation treatments. And doing this, it turned out that the extraction of filters immediately after filtering resulted in surprisingly poor eDNA recovery rates, and that freezing of filters or using a conservation buffer for 8 months resulted in the highest recovery rates. One of the aspects that is, to our view, a must for future eDNA workflows is the inclusion of an internal positive control, which allows valorization and standardization of the protocol that is used. In most of our workflows for both species-specific detection via digital droplet or qPCR or metabarcoding, we are including an internal control at the stage of the filter, prior to extraction, which we measure again after extraction or when the DNA extract was additionally cleaned up. In our lab, we are currently using a plasmide as an internal control, for which we have a good working primer probe set that is compatible for digital droplet, and that we can run in parallel with a large number of our species-specific primer probe sets that we are using. Under optimal conditions, the output of the digital droplet analysis is showing a high recovery of internal control. In our case, in terms of green positive droplets that you can see in the top. The black cloud of droplets are negative droplets, and the pink line is a threshold that we set to distinguish between positive and negative droplets. The internal positive control is an ideal instrument to detect the occurrence of inhibition. When this is the case, this translates into a complete failure of amplification, which you can see in the undiluted reactions of sample 2 and 3, or it can result in reduced amplification, resulting in a lower amplitude of the positive droplets, and this you can see in the twofold diluted reactions of sample 2 and 3 again. The total recovery of the internal control can, on the other hand, be used as an estimate of the extraction efficiency. And both aspects, the detection of the occurrence of inhibition and the measurement of extraction efficiency, helps us to avoid false negative detections. As I said before, this primer probe set for our internal positive control can be run in duplex with another primer probe set targeting or amplifying our focal species or study species, the midwife toad in this example, and that is showing quite similar patterns in this case. In the upcoming slides I want to show you how these estimates of extraction efficiency in combination with the volume of water that is finally filtered can help us to quantify the real eDNA concentrations in the water column more appropriately. To do so, we went out last summer and built up a mesocosm experiment with the larvae of this midwife toad in combination with a large field survey where we took eDNA samples in parallel with intensive netting in order to obtain an estimate of the total amount of larvae that were swimming around in these ponds. If we first look to the uncorrected versus the correct DNA concentrations of midwife toad that we obtained from the mesocosm experiment, we see that we only realize a rather marginal increase in the predictability of the number of larvae that we stocked in these tanks. However, when using the same correction procedure on the field samples, correcting for extraction efficiency and the volume of water that is filtered, we see that we can come up with much better predictions of the total amount of larvae that is swimming around below the water surface when using this correction compared to the uncorrected estimates. Both situations thus clearly show that the use of an internal positive control can offer us much better or stronger predictions of population densities in blind natural systems. Using the same approach for a nature management project last summer, we were not only able to obtain presence-absence data of the Neyrinck toad in a small nature reserve near the port of Antwerp, but additionally tried to offer rough estimates of population densities too. Okay, in the following slides I want to point on the risks of implementing a primer propacer that is not developed in the geographical area where you want to use it. We made that mistake 2 years ago in case of the Oriental weather loach. That is an invasive species coming up from the Netherlands, but was not documented to occur in Belgium until 3 years ago. And the problem with this species is that it is a very successful species that hybridizes with our native habitat directive Miscurnus fossilis and is hanging around in the same habitats. Using a primer propacer that was developed for testing in Australia, it falsely appeared that the Oriental weather loach already occurred at a large number of locations in Belgium. However, and luckily, further investigation of these very surprising eDNA observations revealed shortly after that it were false positive detections that could be attributed to interference of this assay with the closely related Copitistainia, a species that is only occurring in Western Europe. Based on these findings, we decided to develop a new species-specific primer propacer for detecting the Oriental weather loach that is compatible with the primer propacer that we are currently using for the detection of the native Miscurnus officinalis. And both assays can now be used in duplex, resulting in an increased efficiency and lower number of reactions that we need to run for detecting both species in that area. Okay, based on the idea of multiplexing primer probacers for different species, we wondered whether the combination of two target amplicons on different loci can result in a higher detection resolution of a single species. We therefore tested this hypothesis on American bullfrog using two primer probacers. One was developed for bullfrog detection in the USA by Goldberg, and the other one was developed in China by Lin et al. The Goldberg assay is amplifying an amplicon at the cytochrome B area, whereas the Lin assay is amplifying an amplicon at the 16S locus. To not make the same mistake as we did with the Oriental weather loach, we first decided to intensively screen both assays on a large number of natural eDNA samples occurring from ponds harboring all the amphibian species occurring in Belgium, but lacking American bullfrog. And it turned out that they were both specific and safe to use. In the next step, we applied them in simplex and duplex on a number of positive samples to test the effect on detection resolution. To illustrate the output of these tests, we plotted two natural eDNA samples containing bullfrog at variable concentrations. When looking to the simplex reactions, you can clearly see that both assays are resulting in an amplification with positive droplets showing a different amplitude, the so-called digital droplet resolution. Translating these numbers of positive droplets in concentrations nicely shows that duplexing both assays results in a twofold increase of measured bullfrog DNA. When repeating this strategy on a much larger number of samples containing bullfrog at variable concentrations and originating from a larger number of different water bodies showed that these patterns are consistent and further reveals the advantage of using multiple targets at different loci in multiplex. In addition, we also tested in 4 samples that are situated around the limit of detection whether multiplexing at these very low concentrations can result in the reduction of this LOD, which is clearly the case. In other words, multiplexing made us substantially lower the number of replicate reactions needed for accurate detection at very low concentrations. Finally, we also found that multiplexing is reducing the variability among replicated measurements or reactions, expressed in terms of the coefficient of variation, where the threshold of 35 is used for the limit of quantification, and again it appeared that multiplexing substantially reduced this LOQ. We also found very similar patterns in other organisms too, such as the European weatherloach, the otter and also water shrew, indicating that this is a surprisingly overlooked but powerful tool to increase detection resolution in single species tests. The takeaway message of this talk is that the use of multiple markers on different loci in combination with the use of internal positive controls can substantially increase detection rates at very low concentrations and generate more robust and reliable data. Finally, I want to thank all these people for their support and hard labor that is behind these results. And I also want to take the opportunity to invite people to collaborate on new project proposals or other opportunities to work together. Thank you.